Okay, sorry for the delay. Sometimes I get distracted. Okay, so we've been talking about the cytoskeleton, and now you guys have seen all my beautiful movies, and you've decided that you're really interested. Wow, no one is here today. Do you guys hear that? Every time you listen to my lectures, you hear me go, "Nobody's here today." Then you're gonna go to the chair of the department and be like, "See, even she said no one's coming to class." Okay, there really is no one here.
Maybe they still don't have power.
Huh?
Maybe they still don't have power.
Yeah.
They're crazy.
Yeah. Anyways, if they live in New York. Okay. The only people who get any sort of hurricane excuses are people who are commuting from New York City, which is nobody. Or New Jersey. New Jersey. They get it. They'll get it. Okay, so far, okay, this is fun. This is fun. Okay, so far we've talked about the major components of the cytoskeleton: actin, microtubules, intermediate filaments. They're all made through polymerization of monomers, so monomers coming together in some way to form a polymer. The major concepts that we went over that you'll see over and over and over again, and you better know, and if you don't know them, you'll be lost and you'll be studying for the exam crying into your textbook, are nucleation, critical concentration, plus and minus ends, treadmilling, dynamic instability. We went over all those on Wednesday, because everything we're going to talk about is proteins and how they affect these kind of things. So you need to really understand them. And then we spoke about how the assembly and disassembly of the filaments or polymers. So I might call when I mention filament, I mean monomers bound together, or I might say actin filament or microfilament, intermediate filaments, or I might say polymer, which also means monomers bound together. So just so you know. So assembly and disassembly is regulated by NTP hydrolysis, so ATP or GTP, right? For example, the role of the GTP cap in microtubule dynamics. So that's where we are, and now we'll keep going. So here's what we're talking about with the GTP cap. So with microtubules, when they're growing, there's a lag in GTP hydrolysis. Remember, every time a monomer, or actually a dimer in the case of tubulin, every time a tubulin dimer binds to a growing microtubule, you're going to have GTP hydrolysis. But there's a lag, there's a little time lag, which results in this buildup of this GTP cap. So this is a cap of the microtubules, the top part of the microtubules, that contains GTP-bound tubulin. Okay? And it's a cap because it's at the top, right? It's not like there's a GTP put on top of the microtubule. No. It's actually GTP-bound dimers. And when that GTP cap, so when growth starts to slow down, and that's due to the concentration of monomers in the solution or in the cell, that allows the GTP hydrolysis to catch up. So then you have hydrolysis of your GTP cap. You lose your GTP cap, and then you start to disassemble, and you shrink in size, and that's called catastrophe. So treadmilling and dynamic instability, those two concepts, they both aid in the rearrangement of the cytoskeleton. And when we talk about treadmilling, it's that growth on one end while shrinking on the other end, which you see a lot in actin. And dynamic instability is that growth and shrinkage, right? So they aid in the rearrangement of the cytoskeleton. Well, why would you want to rearrange your cytoskeleton? What's the point? If you're moving, right? So if you're a cell and you're moving, you would want to rearrange what component of the cytoskeleton? Actin, tubulin, intermediate filaments. Actin, right? So why else might you want to rearrange your cytoskeleton? So that's right. Yeah. If you're dividing, right? You're going to make mitotic spindles. You're going to move your actin to form your contractile ring for cytokinesis, right? Why else might you want to rearrange your cytoskeleton? Yep. Endocytosis, right. Why not? Because you definitely don't want actin pushing your membrane out when you're trying to do endocytosis. Absolutely. Okay, so now you're understanding that the cell is this dynamic being, which is always changing. So cytoskeleton cannot be static, right? It can't just be one way. Otherwise, the cell would not be able to respond to its environment. Okay. So actin and microtubules, they switch between growth and shrinkage every few seconds for actin or minutes for tubulin. So this is rapid changing in the status of, that's why it's dynamic, right? The cell, it needs to use a lot of NTP to keep this constant, to keep a constant concentration of filaments, right? Remember, you get GTP hydrolysis every time you attach a monomer and dimer. So that seems like a waste of energy, doesn't it? So what's the advantage of doing this? Why would you want to constantly assemble and disassemble, assemble and disassemble? Why not just constantly make new proteins that are these long proteins that stay there and degrade them? I just answered it for you. Yes. You want to raise your hand?
More energy to make the proteins.
Right, so it's going to use more energy to make proteins, right? It's kind of like the question we had about modifying a protein versus making more protein, right? So if you phosphorylate a protein, that takes like no energy, right? Versus transcription and translation of a new protein, right? So this is kind of a way of modifying the structure of a protein. You've got all the building blocks, you got all the bricks, so you just make a building, take it down, recycle those bricks, make another building, take it down. Better than going back to like the clay and making the bricks and molding the bricks and then building it and then taking it down, making it into sand or whatever, dirt, clay, adding the water, churning the butter. I don't know. Yes?
Is it also to keep it from interfering with other stuff in the cell? So you only have a limited amount of space, so you try to keep it functioning in one section.
So you're saying to keep these specific components from interfering?
Yes.
Okay, so what I'm hearing, to clarify, is to keep the different components of the cytoskeleton from interfering with other components of the cell. You have a limited amount of space. Okay, so I want to add to that and say that makes sense in some ways, right? Because you only have so much space in your cell, and if you've got a certain amount of actin monomer, right? And you need, let's say your cell and you're moving this way, so you're going to be growing your actin out this way. Suddenly, your chemoattractant's over there, and you need to turn around and go this way. It makes more sense to disassemble, send your monomers across the cell, and grow this way than to like make new proteins and send them to the right place and all that, right? Does it make sense? Does anyone know what I'm talking about? Who wants to drop the class? You only have a few days left. Feel free. Less exams to grade. No, I love you guys now. You're like my family. My children. Okay. Anyways. Okay, so it's just much more efficient to disassemble and move things around than to constantly make new. Okay. So this is just about that concept of this constant turnover. It's providing flexibility and reorganization of the cytoskeleton. And you're reorganizing your cytoskeleton because you have to meet the needs of the cell, right? The cell's responding to its environment. That's the whole theme of the class, right? The cell's doing what it needs to do, be it making proteins, be it expressing genes, be it signaling, whatever it needs to do, including reorganizing its cytoskeleton in response to the environment in some way, right? So this is just showing just the growth and shrinkage of microtubules over time. So they labeled so they have 1, 2, 3, 4 different microtubules. Is this microtubules? I believe so. labeled, and they're doing it over time. So 0, 125, 07, 669. Strange numbers, huh? And you can see 1 is here, then it's there, then it's there, then it's there. This is a pretty short amount of time, right? So it's constantly growing and shrinking. And so the subunits of the monomers diffuse through the cell rapidly, and this allows them to constantly go to where they're needed in order to respond to the needs of the cells. And so I'm bringing all this up because you're going to really embrace and understand these concepts, because we're going to learn about these different proteins that are going to control whether or not something's going to grow or shrink. Okay. So think about actin. So I've told you about how actin and microtubules, they're dynamic and they're growing and they're shrinking. So think about how they might be controlled differently in different types of cells. Right? So compare a neuron to a macrophage. Right? Think about okay, actin. We talk a lot about cell division and cell movement with actin. Well, is there going to be a difference in how actin is regulated in a neuron versus a macrophage? Do you guys know what a macrophage is? Yeah, white blood cell. So do neurons divide a lot? No. Do neurons move around a lot? No. Do macrophages divide a lot? They divide. Do they move around a lot? Yes. Yeah, so right there you can start to really think about how they're going to be controlled. The at least actin is going to be quite different. So microtubules, think about the role of microtubules, right? So microtubules are involved in what? Transport of things, okay. So vesicular transport. So is that going to be different in neurons and macrophages? In a way, but not, I mean, maybe not in frequency, but maybe in distance, right? Are neurons differently shaped from macrophages? Do they have these long things that things need to travel down? I don't know. I don't know anything about the brain. But I do know that they have these axons, right? Remember? So you need to send things, right? So you need microtubules to persist for long periods of time. Okay. What else? Cytokinesis, not cytokinesis. Mitosis, right? Cell division. Again, that's going to be a difference in neurons versus macrophages, right? So now that I've convinced you guys that there are differences in different cell types, what do you think controls these differences? I mean, in terms of cytoskeletal, right? So how come in a neuron you're not going to have this constant actin rearrangement and this constant microtubule rearrangement? What's controlling that? What determines whether you're going to have actin moving over here and growing or moving over there and growing? What controls whether you're going to have a mitotic spindle form? Yes?
Different regulatory proteins.
Right, very good. Different regulatory proteins, right? Okay, does anyone want to add to that? Right, yep. Extracellular signaling. So does extracellular signaling affect the function of regulatory proteins? Yes. So the two of you together are bringing together different parts of this class, right? So yeah, so you'll have a signal from outside the cell that might say, hey, T cell, time to divide. Signaling. Then cell cycle. Cell cycle activates kinases and stuff like that that are going to then affect things like mitotic spindle formation. Let's see where we're going. It's all related, right? Okay. What else do you think? So we're saying regulatory proteins control these differences. What controls whether or not those regulatory proteins are there? Signaling, some kind of signaling. So gene expression, right? So genes are expressed at different levels in different cells. Let's bring it all together. Big picture. Huge picture. Cells. Large cell has DNA. Genes are expressed or they're not in different cells. Genes are expressed at different levels. We're trying to bring it all back together. Okay. Okay. So we're gonna dive more into that soon. Okay. So, actin and tubulin are highly conserved. So there are many different types of actin in the cell and different types of tubulin in the cell. And not only that, there are different among different species. Actin and tubulin is conserved. So this large network here, is from a study that was done in yeast, looking at protein interactions, and they were looking at proteins that actin interacts with. So every line, so here's your actin, and every line that's drawn means that there's a protein that they found that actin interact. This is how science is done now. These high throughput screens, you're gonna find like 50 million proteins that interact with whatever you're looking at, and they're color coded based on function. So basically, what we're seeing here is actin is involved in proteins that are involved in. Actin is involved in processes from everything from things involved with cell division and endocytosis. We heard that over there. To lipid synthesis. I didn't know about that. And so understanding that actin is involved in all of these different processes, what does that make you think about when you think about how actin is highly conserved? So if it's conserved, what does that mean? What does that mean? Are we talking about conservation biology? No. Okay. Yes. And it stayed the same in what way? So despite evolution, this protein stayed the same in what way? What's the same about it? Sequence. Okay, so same protein sequence, let's say. Why not? Same shape, right? Okay, so if you think about how these, how actin has the same sequence across all these species, and even within the cell, all the different actins have similar sequence. And I tell you it interacts with all these proteins. Does it give you a reason why conservation is really important? Are you making any kind of connections? Yes.
Because it sort of acts like the skeleton key for all these processes.
Acts like the skeleton key. That's very, very seasonal. Skeleton key for all these processes. Yes, right. So who wants to expand on that? I like that thought. What do you mean skeleton key? Are we talking about Halloween? Right, what's a skeleton key? Universal key. Right, so actin's involved in all these things, and then there are proteins that regulate the function of actin, whether actin's going to grow or not, whether actin's going to go here, go there, be stable, or be not stable. So if you're going to have all these proteins that are going to interact with actin, then it needs to be conserved because proteins interact through, what do they interact through? They interact through domains. They interact through the biochemical properties of their amino acids. So if actin wasn't conserved and had vastly different sequences, even within a species, right? Let's say we stay in a species, there are 10, million types of actins. If it wasn't conserved, would you be able to use all these proteins for all these different functions, or would you suddenly need 10 times as many proteins, right? Are you guys following me, or are you guys, what the hell is she talking about? Okay, I saw some nods. I'm going to go with it since no one's raising their hand and crying. Okay, crying? Anyone? No? Does anyone want to cry? Okay. Okay, so that was my point. It's highly conserved because it has to interact with all these proteins that regulate its function. There you go. Okay, so now let's go away from actin and tubulin for a minute, and we'll pay some attention to the intermediate filaments. So actin and tubulin are common to all eukaryotic cells, and as we'll learn later, there are homologs in bacteria. Intermediate filaments are only present in vertebrates, nematodes, and mollusks. They are not required in all cell types. They're only in cells subject to mechanical stress. Makes sense, right? In squishy animals. So if you are super hard animal, who is an ecologist in here? Anyone? Someone is. Yes. Okay. Good. So you can think about this maybe at a deeper level. If you're not squishy, do you need proteins that are gonna keep you from your cells from coming apart? No. I mean, because it's all about resistance to mechanical stress. Does that make sense to you guys? So it makes sense that they're only in those types of animals. Okay, so intermediate filaments, they're constructed from subunits made of monomers. So there's a theme here. Subunits come together, make filaments, right? Subunits made of monomers, and in this case, they dimerize. So here's your monomer, and they dimerize to make this coil-coil dimer. And they associate with other dimers in a staggered way, and they form a tetramer. So there's your tetramer, and then the tetramers will pack together. And what you get is this kind of rope-like structure, and that's where all the strength comes from. There's no NTP binding involved, not like actin. Are you guys sensing already ways to compare things? Right? Okay, good. I saw some nods. No NTP binding site. There's no polarity in this case, and you're going to see that there's no dynamic instability when it comes to intermediate filaments. Would you want dynamic instability in the things that like give you resistance to tend to stress, like to physical stress? Do you want dynamic instability in the thing that's protecting you from the outside world? Okay. So 8 tetramers pack together and they form a rope-like filament that looks like this, right? There you go. It easily bends. It's difficult to break, just like a rope, right? There are many types of actin and tubulin, which I've kind of referred to, but we haven't gotten into great detail. But there are many types of actin and tubulin, and they can be cell specific. There are also different types of intermediate filaments with different functions. So some are nuclear, right? So they're located in different places. Some are nuclear, some are in muscles only, some are only in glial cells, neurons, epithelial cells. Just understand that. Don't memorize that, please, or else I kill you. So we talk about how there are different types of keratins, different types of intermediate filaments. An example is keratin, okay? So there's 20 types of keratin proteins. It's a type of intermediate filament just found in human epithelial cells. There's the staining. There's where your keratin is. And they're cell type specific. And they're made up of heterodimers of 2 types of keratin chains. Remember we talked 2 seconds ago about how there are these monomers that dimerize. So in the case of keratin, they're heterodimers. And the diversity in keratins is useful. So it's useful in diagnosis of epithelial cancers because people know that this type of keratin is in these kind of cells, this type of keratin in these kind of cells, so it's helpful in diagnosis. They provide mechanical strength to epithelial tissues. They actually anchor intermediate filaments at sites of cell-to-cell contact. So those are the desmosomes, and at sites of cell matrix contact, which is hemidesmosomes. And so right here what you're seeing is when there's a defect in the keratin gene, in one of the keratin genes, it results in this kind of blistering because they're not interacting well. And I'll show you another. So here this is called epidermolysis bullosa simplex, and it's a blistering skin disease. And so normally your skin looks like this with the different layers, right? And when you have a defect or a mutation in one of your keratin genes, it results in a lack of interaction. So these keratins are expressed in the basal layer of the epidermis, and so that's over here. And so it's just basically causing the skin layer to kind of come up, and that's what's causing the blistering. So it's important for keeping your cells together. Other defects with intermediate filaments. This is specific to neurofilaments. ALS or Lou Gehrig's disease is associated with accumulation and abnormal assembly of neurofilaments. So this is a family of intermediate filaments that's found along axons in neurons, and the defect, they think, interferes with normal axonal transport. So these are neurofilaments, and these are glial filaments. And here's an axon cross-section. And so here, these are microtubules, but all these regularly, it's pretty regular the way they're organized. These are all neurofilaments. And so when you have a defect in that, it's resulting in, it's preventing normal axonal transport. Okay, so let's go back to actin and tubulin. Any questions so far? So actin and tubulin are frequently targets of toxins, right? If you were producing a toxin, wouldn't it make sense to produce the toxin against actin or tubulin in some way? That's a really great way to be toxic, right? Because I just told you about how important actin and tubulin are in everything. So if you're going to affect whether or not actin can assemble or disassemble, or whether or not tubulin can assemble or disassemble, that'd be a really great way to be toxic. Some examples is latrunculin, which is from a sea sponge. It binds actin monomers, preventing assembly. So what would be the result of preventing actin monomer assembly into actin filaments? How would that affect the cell function? Yes? No cell movement, right? Okay, no cell movement. What else?
Cell division.
No cell division. Would you say that'd be bad? Is that good or bad? Right? Okay. Phalloidin, which is actually used in the lab to stain actin. So sometimes when you see immunofluorescence pictures and it's red, they'll actually color the phalloidin red, and then when you add it to your cells, it'll bind to actin because it binds and stabilizes actin filaments. So if you stabilize your actin filament, is that good or bad? Come on. Bad, right? We talked about how important the dynamic instability properties. So stabilizing or preventing assembly, both are bad because you're killing any ability to assemble and disassemble, right? Right? God. I quit. Okay. So that's from a fungus. Then there's colchicine, which is from the autumn crocus. And it binds to and stabilizes free tubulin. What do you think the outcome of that is? You can say good or bad. How's that? We're going to go scientific here. Good or bad? Bad, thank you. Right? So if you're binding and stabilizing free tubulin, are you able to polymerize and become a microtubule that's growing? No. No. So, so this is useful, right? Okay, so these are toxins, but can you see how this might be useful for drug development? What kind of, let's say you were who wants to go to pharmacy school? Okay, so these are the people who should be super interested right now. Who wants to go to pharmacy school and finds this horribly boring? Okay, good. Good. Okay, so you're not only going to pharmacy school, but now you're just a chemist, and you're creating drugs, and you want to create a drug for something, and you see these, and what do you think of automatically when you see these different toxins? I want to make a drug that for this disease. What are you thinking? What did I hear? Cancer. Yay, cancer. Yay, cancer. Right. Because if you do any of these things, especially anything that's going to affect tubulin, that's great for a cancer drug. Because what are cancer cells doing all of the time? Dividing. So if you prevented mitotic spindle function, that'd be a great idea. So And it turns out that's already been done. So we're not that smart. Those before us were smart. So Taxol, who's heard of Taxol? Yeah. So that's from the bark of the Pacific yew tree. I love when they tell you where these things are coming from, and it sounds so like, I feel like I'm going on a little vacation. So we should do a class vacation to go find some Pacific yew trees. Yeah? Okay. I see people in agreement. So Taxol binds and stabilizes microtubules, and it's used to treat breast and lung cancer and preferentially kills dividing cells. Why preferentially kill dividing cells? Why doesn't it kill all your cells? Well, then you die. Well, yeah. Yes. But like, have you ever thought about why? Like, so for example, why when you're giving someone chemo, why it doesn't kill them, right? Because you're killing their cells. But what it does is it preferentially targets dividing cells, which are the cancer cells, right? Because, for example, this one is going to affect mitotic spindle assembly and function and induce apoptosis. And while it's going to hit the rapidly dividing cells first, right? Because they need that mitotic spindle more frequently. That makes sense? So this is a picture of HeLa cells. Who knows what HeLa cells are? Who's taking Dr. Tsuji's class? So then you guys really know what HeLa cells are. So these are HeLa cells from Miss Henrietta Lacks. Poor Henrietta Lacks, right? Poor Henrietta. No, Henrietta had no idea what was going on, but her family found out that her cells were at Hopkins and had no idea of what cell lines were and thought that their poor mother was like in a freezer somewhere at Hopkins. Very sad. But anyways. These cells, though, are used in the lab constantly. They're very useful. I use them all the time. They're cervical cancer cells. So these are HeLa cells at metaphase. See those chromosomes lined up? What do you think the red is? Right. So microtubules, right? And so these cells were treated with Taxol, and as you see, the chromosomes fail to attach. So here's some chromosome down here. Here's something over here. They fail to attach in the mitotic spindle appropriately, and that results in cytokinesis failure. And as we'll see as we get to cell cycle and we talk about mitosis, there are all these checkpoints. Remember learning about checkpoints? And usually, if a checkpoint is activated, what happens? Cell cycle arrest, right? So these are things that are good when you're treating someone for cancer. You want to arrest the cell cycle. You want to promote apoptosis, right? Okay, so that's how one cancer drug works. It binds and stabilizes microtubules. Questions? Okay, so let's talk about bacteria. I don't talk about bacteria much in this class, but for some reason I decided to here. So they have a tubulin homolog. They don't have tubulin. It's called FTSZ, and it's similar in structure, has an kind of like an alpha beta tubulin. It also hydrolyzes GTP during polymerization. It also has it's also dynamic. So its half-life is about 30 seconds. And it's involved in formation of the Z ring at the septum during cell division. So does anyone understand how bacteria divide? Does everyone understand that? It's a little bit simpler than how we divide. I'm a cell. A little bit simpler. So they have a septum that forms, and then the two cells come apart. And so you can think of this Z ring is similar to the contractile ring during cytokinesis, except for the fact that this is formed by a tubulin homolog, and the contractile ring is formed by actin. So totally different, but similar function. And it also rearranges rapidly during the bacterial cell cycle. And so you could see time 0 all the way to 37 minutes. And so here's your FTSZ staining, and you can see it's pinching off, and then you've got your division. And then it actually is already assembling over here for the next one. So it's similar to the contractile ring, but remember, it's not made of actin. And here's another one, dividing of chloroplasts from a red algae using a protein ring made from FTSZ also. And so you can see here it's stained yellow, and it's pinching and separating. So then there's also a bacterial homologue of actin, well, two of them, MREB and MBL, and they play a role in cell shape and chromosome segregation. And so this is just an experiment where they have wild type MBL and MBL deficient mutant bacteria. And so you can see here just this normal rod-like shape, and then you have cell division happening. And here, the shape is all messed up, and they're not dividing; they're getting stuck together. So that's how you do. That's the nice thing about working with yeast and bacteria is you can do these knockouts and look for nice, especially when you're working on cell cycle. There's a lot of nice phenotypes you could just visibly look for. Okay, here's another homologue. Do not memorize all these. Just appreciate them. Acknowledge their existence. Appreciate that these functions are conserved from bacteria all the way through us in some way. So this is another homologue of actin that's involved in plasmid segregation. So do you guys know what a plasmid is in bacteria? What's frequent? So what is a plasmid? Someone tell me. Yes.
It's basically a little bubble sort of extra DNA.
So it's a little piece of extra circular DNA. Yep. And what kind of genes are usually encoded on plasmids? Yes.
Drug resistance?
So drug resistance genes. What else? Anything else? Virulence genes sometimes. Yep. And plasmids are nice because they can kind of be shared very nicely between bacteria, sometimes of the same species, sometimes between species, which is really scary when you start to think about drug resistance, right? Anyways, so this part M is responsible for plasmid segregation. So in this picture, they're showing you've got these part M monomers, and they also assemble into polymers, and they're interacting with some kind of protein that binds to the origin of replication. So there's your plasmid. This red part's your origin of replication. There's some kind of protein there that binds to part M, and it pushes that apart, and then your septum's going to develop here, and you'll end up with two bacteria with plasmid on each side. And so this gene is specific to plasmid segregation. So where do you think the gene might be located? Do you think it's on the bacterial chromosome or on the plasmid? Just for kicks. Where do you think it might be? Okay, well, for those who know things about plasmids, usually plasmids tend to encode all the genes required for the genes in that plasmid to function, including segregation of the plasmid to daughter cells. So usually the part M gene is also on the plasmid. Okay, here's another homologue. So this is an intermediate filament homologue called crescentin, and it's in Caulobacter crescentus, and it's responsible for a crescent shape of the bacteria. So here's the bacteria, and it kind of creates this line on the side. So this is staining for crescentin, and it's just involved in the sickle-like shape of the bacteria. And so here's mutant, here's wild type, and here's mutant. So you can see it's clearly responsible for the shape of the bacteria. Okay, so what is the relevance of these homologues? Why am I telling you about this? So we have something to talk about on a Friday morning? I could have come up with many other things to talk about. I could talk for days. I have that gift. Well, I just want you guys to acknowledge the fact that there are homologues, and when you have homologues across many species and many kingdoms, that says something about how important genes of that function are, right? It says something about importance. If you have one gene that's only present in one species of one bacteria, is it really important to all the bacteria in the world? No, right? Otherwise, it would be in all of them. Okay. So you might, when you're thinking about this, I want you to think about how the functions of these different proteins are similar or different from their eukaryotic homologs. And there's one function that is missing, right? So we've talked about the different functions of actin and tubulin and intermediate filaments, right? We've talked about functions in shape. We've talked about functions in plasmid segregation or chromosome segregation, right? In cell division. What's one function that we learned about with the eukaryotic homologs that we have not yet seen in bacteria? A function of a cytoskeletal protein called tubulin. Yes? Movement. Movement of what? Movement of the cell. Well, I was going for movement of something else. Movement of something inside the cell. Remember, we talked about vesicular transport, right? So we haven't seen any examples of vesicular transport in bacteria. Why?
No membrane-bound organelles.
No membrane. That's a trick question, people. No membrane-bound organelles, right? Are there is there vesicular transport in E. coli? No. Not that I know of. I don't know. We'll have to ask Dr. May, the resident microbiologist. No. And so then we also haven't seen motor proteins, right? There's no homologs of motor proteins. And that also has to do with the fact that bacteria, they do not have vesicular transport. They do not have, motor proteins are used in the mitotic spindle. They're not doing that way. It's just a simpler machine. Okay. Oh yeah, we can keep going. So, the cell needs to regulate. Remember my favorite word. What are my favorite words? Regulate, mechanism, and ubiquitin. Are those my favorite words? Right? The cell needs to regulate the length and stability of its cytoskeletal filaments, right? So how does it do this? There are these accessory proteins, and they bind to filaments or the free subunits, and they modify dynamics or structure. And I'm going to direct you to panel 16.3 in Alberts for a good illustration of the kinds of proteins and their functions. Look at it, love it, think about it. It's just, I like pictures. It's a nice picture. Okay. And the reason I didn't put it up here is because for some reason I can't get access to the figure to put it in the PowerPoint. So I'm going to just tell you to look at it. Okay. That's all. No, I'm just kidding. Okay, so the first category of proteins that we're going to talk about are proteins that nucleate filaments. So what does that mean to nucleate a filament? I'm going to do a dance until someone tells me something. Remember we talked about nucleation. Yes? Bring the initial subunit together. Remember there's this thing that we did where there was this like that, looked like that, and here's like there's this binding of monomers together, and then here you get the nucleus formation, and then you get this growth, right, until you hit your steady state, and that's like your critical concentration, right? Something like that. It looked like a bacterial growth curve, but had nothing to do with bacteria. Okay, so proteins that nucleate filaments. They actually bypass this part, and it's almost like you're starting right here, right? So how would this regulate microtubules? If you have a protein that's going to nucleate a microtubule, is it going to cause it to grow or shrink? Grow. And how about actin? Is it going to cause it to grow or shrink? So proteins that nucleate filaments tend to encourage growth, okay? That's a good way to think about it. Okay, so with microtubules, gamma tubulin is involved in nucleation. And the gamma tubulin is part of the gamma tubulin ring complex, gamma Turk. I like to call it gamma Turk. And that is present in the centrosome. We'll get back to that in a minute. Okay, so here's how it works. You've got your microtubules. You've seen that, your alpha beta microtubulin. And then you've got your gamma tubulin and some accessory proteins that are in the gamma tubulin ring complex, and that forms the nucleus, upon which your dimers, tubulin dimers, quickly assemble, and you have this rapid growth. Okay, this is the last slide, and then you can go away. So in animals, a microtubule organizing center, MTOC, located near the nucleus, it's called the centrosome. It contains more than 50 gamma Turks. Okay, so you've got this circular thing, this centrosome, and it's got tons and tons of these nucleating proteins, these nucleating gamma tubulin ring complexes, which will nucleate microtubule growth. Remember, we looked at all these pictures of interphase cells where you've got your microtubules radiating out of the center. That's what is doing it. That's what's causing it. So the microtubules organize here, and they push outwards against the cell membrane, creating the centrosome. And they're pushing outwards, and that's why it's a centrosome, because if you've got pushing outwards in every direction, it's going to center your centrosome, right? Kind of. They duplicate during interphase, right? And they move to opposite sides, right? Because what they're going to become the mitotic spindles. Okay, we're going to stop here, and I'll let you go. Happy weekend.